Okay, we now move to the second presentation and we leave discussion at the end. So I'm happy to call on the stage Valerie Crowley from University College London, who will give us a talk about the biomarkers in the BILCAP series with a specific focus on HER2.
So, hi everyone, it's a pleasure to come to AMMF and I'm very grateful to be invited to talk. So my name is Valerie Crowley and I work at the University College London Cancer Institute. And so today I was going to focus on some of the results of our translational analysis from the BILCAP clinical trial with a particular focus on a molecule called HER2. So I think we've had enough background already sort of over the course of the conference on cholangiocarcinoma. But one of the interesting facts about cholangiocarcinoma that I think came up a lot in particular yesterday is that the mainstay of treatment at the moment is still mainly chemotherapy, both in the adjuvant and the metastatic setting. Obviously, now we have pemigatinib that's been licensed for FGFR2 fusion patients, but for the most part, most patients who are treated for cholangiocarcinoma are going to receive chemotherapy. And while chemotherapy in itself isn't so much of a terrible treatment, a lot of patients simply aren't well enough to have chemotherapy. Or if they do have chemotherapy, unfortunately, their cancers, they don't respond to treatment and they get more and more unwell. So there really is a need for more sort of focused, more targeted treatments that are more individualized and personalized to individual patients. And in order to find these new targeted treatments to sort of increase patient response to treatment and reduce side effects, we need to have a better understanding of which cancer driver genes are most important in biliary tract cancer and cholangiocarcinoma. And even more importantly than that, as well as knowing what the incidence and prevalence of these genes, mutations, and alterations are, we need to know what effect that these alterations are having on patient survival and on whether or not patients relapse after surgery, because that's something that is quite difficult to investigate. So the BILCAP clinical trial was a very large phase 3 clinical trial that recruited patients between 2006 and 2014. And it recruited patients who were operable, so who'd had surgery, so all had early stage cancer. And patients were randomized in a 1-to-1 fashion to either have chemotherapy after surgery, so they had a drug called capecitabine in the adjuvant setting, or observation alone, which was the standard of care treatment at that time. And a total of 447 patients were recruited, which, cholangiocarcinoma and biliary tract cancers are generally quite rare. So to recruit this number of patients for one trial is a very large number. And in the end, when the overall analysis happened, it was seen that the patients who were given chemotherapy did better than those who were not, which is why the standard of care here in the UK is that patients who have surgery have chemotherapy afterwards. And I think the important thing and the reason why we're focusing so much on the BILCAP clinical trial is there are quite a lot of genetic databases out there, so things like the European Genome Phenome Archive and things like the 100,000 Cancer Genomes Project. But the issue with a lot of those is, A, there aren't that many biliary tract cancer patients in even these huge cancer databases. But even when you do have the genetic and genomic information, there, for the most part, very little clinical information associated with it. So you might be able to find, for example, whether or not a specific case had a specific mutation, but other than perhaps age and gender, you wouldn't be able to find out their survival, whether they had surgery, did they have chemotherapy. That information is just lacking. Whereas being able to perform the genomic analysis on clinical trial data means that you're able to have the clinical information together with the genomic information. So we decided in particular to focus on a molecule called HER2, or encoded by the ERBB2 gene. And HER2 is a receptor tyrosine kinase that's found on the surface of basically all cells. And what it does is when its ligand binds to it, it binds either to another molecule of HER2 or to a related molecule, and that binding together, that dimerization, causes the activation of various downstream pathways. And those downstream pathways are the ones that are generally involved in cell proliferation and anti-apoptotic pathways to stop cells from dying. And amplification, so having more copies than is normal of HER2, or having mutations that, for example, activate HER2 without ligand binding or without the need for a dimerization, are pretty common in a variety of very common tumor types. So we know, for example, that breast cancer, stomach cancer, esophageal cancer, and lung cancer often have HER2 alterations. And that means that there are well-known and well-licensed drugs that target tumors which have HER2 alterations, amplifications, and one of the most well-known ones being trastuzumab or Herceptin. So what we did is we collected together all of the FFP or fixed formalin blocks from the patients who'd recruited and treated on BILCAP. We collected all of those together, and they went forward for DNA and RNA extraction. And once we'd extracted the DNA and RNA, we performed some various genomic tests on it. So we managed to carry out targeted gene panel sequencing to look in particular at gene mutations. We performed what we call whole genome sequencing, but very sort of shallow, low whole genome sequencing to look at alterations in copy numbers of the specific genes or any mass sort of chromosome rearrangements as well. And we also managed to look at RNA sequencing to look at gene expression and to have a bit of a look at the tumor microenvironment as well. And then we used bioinformatics analysis to figure out what was going on from that information. So at this time point of the analysis, we looked at the total of 45 intrahepatic cholangiocarcinoma patients. So 45 of them had the low-pass whole genome sequencing and the RNA-seq, and 36 patients of those 45 had the in-depth targeted sequencing panel carried out. And overall, the types of gene alterations and mutations that we found were very similar to those found in similar data sets. Often data sets are a lot smaller, but similar data sets. So, for example, we found that 9 of our 45 patients had a fusion in an FGFR gene, so around 20%, which, if you look at the sort of scientific literature, is around the type of number that we would expect. We also, at a lot lower rate, found fusions in NTRK1, in FGFR1, and in FGFR3. We found a lot of mutations in various cancer driver genes. They tended mostly to be what we call variants of unknown significance, so not mutations that we know are particularly oncogenic, but finding definite mutations in known oncogenic genes. So we found quite commonly mutations in ROS1, in MET, and in ALK. And when we looked in particular into pathogenic mutations, we found, again, sort of the number that you'd expect of mutations in IDH1 and mutations in FGFR2. And then when we looked at amplified genes, we found that NTRK1 was relatively commonly amplified. And we did found that 18% of patients had a HER2 amplification, and as well as amplified MET. And we looked a little bit into the tumor microenvironment just to see how many patients had particularly high or low levels of regulatory T cells or CD4 T cells. And we found there was a small but significant proportions who seemed to have quite an inflamed tumor microenvironment compared to what you might expect. So, but the main point of our study was not just to look at the incidence and prevalence, because that's been looked at before, but to see what difference any of these made to patients' overall and progression-free survival on treatment. And basically, we found that most of the alterations we were looking at didn't really make a significant difference as to whether or not people lived longer in general, or whether or not people were more or less likely to have their disease relapse after surgery. So when we looked at overall survival, as you can see sort of from the forest plot here, most of the alterations didn't have a significant difference on survival. So in particular, for example, with the FGFR2 fusions, simply having an FGFR2 fusion didn't really make a significant difference as to whether or not you were going to be a long-term survivor or not, which some of the smaller studies that had been looking into it perhaps suggested that there might be a difference, but when you started looking at big enough numbers, the significance just disappeared. And, and other things, so we looked at different sort of tumor cell, immune cell fractions, and again, most of those didn't make a significant difference either. And a lot of the other fusions and amplifications, and we looked at HER2 amplification as well, and that didn't make a big difference. And it was a similar situation. We looked at progression-free survival. This hadn't really been looked into before because it's unusual to have a data set where you have both the genomic data and information on whether or not patients actually relapsed after your sample had been taken. But again, we found that most of the mutations and alterations that we looked at didn't make a massive difference as to whether or not people's cancer came back after they'd had the operation. So just to look at some of the specific Kaplan-Meier curves, 20% of the patients had an FGFR fusion, which would be about what we would expect, but the curves are basically the same. It didn't really make a massive difference on either survival or on whether or not the tumors relapsed after surgery. And, our focus was on HER2, or looking at ERBB2. We didn't find any actionable HER2 mutations in our 36 patients where we had the targeted gene fusion. Again, not that unexpected because ERBB2 mutations are reasonably uncommon in this tumor type. We estimated a little bit when it came to tumor expression. It's because of the sample type that we had looking at your normal differential gene expression is difficult. But on our estimation of patients who had particularly high expression in total of HER2, we only found 2 of those, and that's not enough people to make any intelligent guesses as to their survival. But when it came to amplification, we definitely found that there wasn't a significant difference when it came to the patients who had amplified ERBB2 compared to normal levels of ERBB2. But we did find one interesting thing. So we did find that patients, so there's a gene called MET, which is important as part of the cell proliferation pathway, and we found that patients who had an amplification in MET, and from a bioinformatics view, we calculated that as copy number greater than 4, it didn't matter if the total ploidy was high or not. And we found that in 7 of our patients. And we found that the patients who had a MET amplification did do significantly worse than those who did not. So, both when it came to overall survival and when it came to their risk of relapse. So, in our particular data set, at least with the 45 that we had at this particular time, we found that patients with amplified MET did worse, were more likely to have a lower overall survival, and were more likely to relapse after an operation compared to those with normal copy numbers of MET. We found a similar thing for FGFR3 fusions, but that was only 2 patients, so not really, again, enough to make an actual statement of that until we have bigger numbers. But what it does mean is it does look like MET and FGFR3 fusions, or potentially other things, may be important indicators and may be easily testable indicators as to whether or not somebody will have a longer or shorter overall survival, or whether or not somebody is more likely to relapse after surgery. And these might be useful in developing a prognostic test in the longer term, and they might well be sort of potential targets that you might want to focus on when it comes to targeted treatments in the future. And again, 45 patients is a reasonable number, but we're getting more and more of those total of 447 coming through, and it might well be that other alterations that don't quite meet significance in this particular analysis may start to reach significance. So we're going to be finding more and more information as it goes on. Like I said, I wrote this presentation last week when we had 45 patients. As of last Saturday, I've now got 98 patients. And, so as the patient numbers expand, the exact genes might change, but we're starting to find more and more patterns as to which genes are important when it comes to affecting overall survival and progression-free survival. And then the next steps as well is we're also in the business of obtaining more genetic samples from other similar large-scale clinical trials. So it's a similar sort of phase 3 adjuvant chemotherapy study was run in Japan called JCOG 1202 or ASCOT, which was comparing sort of adjuvant S1 versus observation. So that's a similar patient cohort, a similar patient group, and also very large numbers of patients. So we'll be able to have at least the RNA sequence genomic data as well as the clinical data from that patient group. And then sort of as an alternate comparison, we're also about to get access to a data set of patients with metastatic, who were metastatic at presentation. So there was a phase 2 clinical trial investigating a drug combination in metastatic patients, and to see whether or not there are any significant differences between patients who present early with resectable cancers, which has been where most of which has been mainly the focus of most of the genomic investigations, and see if that is significantly different to patients who present late or present with metastatic disease. So thank you very much for listening. Just like to say thank you in particular to my supervisor, John Bridgewater, who's not here anymore, and my other supervisor, Javier Herrero, to John Primrose and everyone who worked on BILCAP, and everyone at the UCL BLIC where I work. So thank you very much.